Okay, so just be aware, everyone, that I'm recording. I'm gonna share my screen now, which is hopefully everyone can see it now. Can you guys see it? All right. And then,
sorry, okay.
Can you see it?
Yes, it's not in full, but it's better than before. Now it's full. Thanks.
Yeah. Okay, perfect. So hello, everyone, and good afternoon, good morning, and welcome to the European Society for Marine Biotechnology webinar series for 2022. I am Maria Salta, and I'll be chairing this session along with our president, Concetta, who is with us today, Professor Messina, and along with also Eleonora Curcuraci, who will be giving the talk. I'm going to just briefly introduce you to our society, as I always do. So our society was established in 1995 in April in France, and the main goal was to promote marine biotechnology within Europe and to help with closer research collaborations between different labs and marine biotechnologies. So we want to exchange scientific information, and the goal also is to help with training and cooperation of our members and help the society and the environment by doing all that. What are key activities? Well, we do these conferences every once a year, usually, or every 2 years. Actually, there's one coming in next year, so please keep an eye on this. The details will soon be uploaded on our ESMB main webpage. So we organize webinars like the one you are participating now. We have meeting and training opportunities for, especially for students in early careers. You can have access to the Journal of Marine Biotechnology from Springer. And of course, we have our newsletters that are distributed to all our society members. Now, if you want to have more information, these are some of the venues you can actually access. And this is the ESMB.org. Our Instagram, which is actually handled by Eleonora, who will be giving our talk today, and our LinkedIn, which is also very useful, which is literally European Society for Marine Biotechnology. If I am forgetting something, Eleonora can probably help. I have also a chat at the moment. Okay, yes, no, there is no extra information. And I will then, without any further ado, introduce you to Eleonora Curcuraci, who will be talking to us about the title that is From Universities to Industries. It's the NUTRAQUA project, which is production of high value added nutraceuticals in a multi-trophic aquaculture system within a closed circuit marine hatchery, and it's a doctoral program with industrial innovation. So you can find the bio and the talk abstract in our ESMB website. I want to give the bio of Eleonora, and I will do so at the end because at the moment both of my screens are occupied, so I cannot read it out and I can't remember everything by heart. I'm so sorry, Eleonora, but I will give your bio at the end of your talk because you have a great experience and I really want to share this with the society. So I think at this point I want to stop sharing, and I hope I did that. Yeah, and then welcome, Eleonora.
Thank you.
And I'm just gonna be muting, and please share your screen and you can continue.
Yes, okay. Do you see my screen?
Yes, we do. And yeah, now it's full screen. Perfect.
Okay, perfect. Thank you so much. Good afternoon to everyone. First of all, I would like to thank Professor Concetta Messina, that is the president of the European Society for Marine Biotechnology, for inviting me and giving me the opportunity to present the results of my PhD project. And I also would like to thank all the board members of the society for welcoming me and for guiding me in the board activities that we have done so far. As Maria said, my project, my PhD project was entitled NUTRAQUA, but I also wanted to highlight in this presentation that this project was done from university to industries, since it was a doctoral program in industrial innovation. So my project was an innovative project with an industrial characterization and was linked to two of the areas of the Italian National Strategy for Smart Specialization, that are health, food, quality of life, smart and sustainable industry, energy, and the environment. For each of these areas, there are specific development trajectories that are nutraceuticals, nutrigenomics, and functional foods, and systems and technologies for wastewater treatment. The project was also in line with the European Blue Growth Strategy, which main objective is to develop the aquaculture sector through the support of marine biotechnology. The project was also in line with some of the Sustainable Development Goals that were promoted by the United Nations in 2015, aiming to reduce world poverty and safeguard the planet by implementing a set of targets by 2030. Specifically, the goals that were intercepted by my PhD project were: number 2, aim to reduce hunger in the world; number 3, to guarantee health and well-being for all organisms; number 12, to promote and guarantee responsible production and consumption of resources; number 13, for climate safeguard; and also number 14, for oceans protection. In the recent decades, there was a decline in fishery catches that was driven by poor management of fish stocks and also a simultaneous increasing demand for fish products. This decline has been coupled by the growth of the aquaculture industry, which has expanded all over the world. In the Mediterranean, the aquaculture industry is dominated by monocultures, particularly sea bream and sea bass, although in the recent years a greater species diversification has been reported. It is known that the development of the aquaculture sector must be able to ensure high levels of environmental and social sustainability through the development and establishment of good management practices that have to be efficient and also diversified in terms of products. The sustainability of the aquaculture sector can be ensured by maintaining high quality standards, product traceability, transparency on production condition, and also responsible farming practices and the integrity of aquatic ecosystems. So in this context, integrated multi-trophic aquaculture represents an ideal system that is capable of optimizing energy balances and economic yields through the co-culture of fed species and extractive species that through nutrient recycle ensure the treatment of wastewater from the aquaculture facility and also produce biomass. This biomass is rich of lipids and bioactive compounds that have proven beneficial characteristics for both farmed organisms and also for humans. So in this regard, The main goal of NUTRAQUA project was to link marine biotechnology with the aquaculture sector for the development of integrated multi-trophic systems by applying technologies through the identification and the co-culture of extractive species, the optimization of the production performances by the extraction of high-value nutraceuticals, and by the development of monitoring systems. The expected impacts of the project development were linked to increasing economic and environmental sustainability of this activity, and improving consumers' perception of the aquaculture sector. To achieve these goals, were identified extractive species to be co-cultivated in a multi-trophic aquaculture system. In particular, were selected 5 microalgal strains that were Nanochloropsis, Isochrysis galbana, Feodactylum tricornutum, Chlorella, and Donaliella viridis, 1 macroalga, Gracilaria gracilis, and 1 halophyte, Artrocnemum macrostachium. The project was carried out in close collaboration with the industrial partner that was the society Alge, that was interested in the development of multi-trophic aquaculture systems within the Azienda Ittica San Giorgio, that it's a land-based recirculating aquaculture system hatchery of sea bass and sea bream located in Sicily. So over the course of my PhD studies, what I did was cultivating the microalgal strains, modulating the physical and chemical parameters of the cultures in order to optimize the global and lipid yields. On macroalgae, on the macroalga and on the halophyte, I carried out antioxidant quantification and also assessed the antioxidant power of the extracts obtained. On the microalga Feodactylum tricornutum, I performed gene expression analysis to assess the effect of culture conditions on genes that are associated with lipid biosynthesis and reducing power. And finally, I evaluated the bioactivity of the extracts in vitro to assess their protective capacity against oxidative stress on fish and human cell lines. And I also assessed the bactericidal activity of the obtained extracts. The microalgal strains Nanochloropsis and Isochrysis galbana were selected because they are already widely used in the aquaculture sector, while Feodactylum tricornutum was selected since it is considered a model organism in the biotechnology sector. Those strains were maintained under specific culture conditions, and in particular, Feodactylum tricornutum was maintained both under standard nitrogen condition and under starvation nitrogen condition. Lipid yields show that the ability of microalgae to produce lipids is species specific, but it is also related to the different culture conditions that microalgae were subjected to. And this ability to direct metabolism towards a lipid or antioxidant production in microalgae, depending on the species or depending on the different culture conditions, was confirmed by the analysis of total polyphenols and total carotenoids. So therefore, through the modulation of the culture media, I optimized the lipid and antioxidant yields in these microalgal strains as a preliminary tool for the scale up of their co-cultivation from the laboratory to the industrial scale. As I said before, since Feodactylum tricornutum is considered a model organism that responds to nutrient imbalances that are such those that can occur in an aquaculture facility. On this diatom that was grown both under standard and starvation conditions in association with lipid and antioxidant yields, I also monitored the expression of specific genes that are associated with lipid biosynthesis and on the modulation of reducing power. This experiment highlighted that different culture conditions led to different modulations of the associated genes. So the use of molecular references markers can be considered as a useful tool for monitoring cultures in the laboratory. A further experiment was conducted on 3 indigenous Sicilian microalgae strains that were isolated in Pozzillo, Barcarello, and in Trapani salt ponds. The aim of this experiment was to identify local microalgae strains to be used in the development of Sicilian outdoor aquaculture, which must take into account the high temperatures that can be reached in summer in Sicily. Therefore, the 3 microalgae strains were cultured under different salt and temperature conditions to evaluate their productive performances in lipids and antioxidants. The results showed that the highest lipid yields were obtained by Dunaliella viridis, particularly for omega-3 fatty acids. Furthermore, the low temperature condition led to an increase in lipids in Chlorella Barcarello, particularly for omega-3 fatty acids. With regard to antioxidant production, the high temperature and the low salinity led to an increase in polyphenol, in chlorophyll, and in beta-carotene yields in Dunaliella viridis, while the low temperature condition increased beta-carotene yields in Chlorella Pozzillo and also in Chlorella Barcarello. This study highlighted the best conditions for optimizing production yields in lipids and antioxidants in these microalgae strains for their use in the development of Sicilian regional outdoor aquaculture facilities. A further study was conducted on the macroalga Gracilaria gracilis, which was cultivated within the hatchery pond, and on the allophilic plant Artrocnemum macrostachyium, which grew spontaneously along the hatchery pond. The aim of this experiment was to assess the polyphenol yields of this macroalga and this alophyte in relation to the specific growing conditions that are peculiar of the hatchery pond. So yields were evaluated by comparing extraction techniques using organic solvents and an innovative green extraction technique, that is the supercritical fluid extraction, which does not require the use of solvents and has a short extraction time. The results show that although the yields obtained by SFE were the lowest, the antioxidant power of the extracts obtained by supercritical fluid extraction was the highest. This study revealed that these matrices cultivated under the specific growing conditions within the hatchery pond represent an important source of natural antioxidants to be used in the nutraceutical and cosmeceutical sectors, especially since the polyphenols that were obtained using a green extraction technique. But as the use of bioactive compounds in the nutraceutical and cosmeceutical fields require a great number of analyses. Another experimental step was aimed to verify the bioactivity of the extracts obtained and to assess their protective effect against an oxidative stress. In particular, human fibroblasts were pre-treated with Arthrocnemum macrostrachium extracts and then induced to oxidative stress through UV radiation. The pre-treatment of cells with the extracts obtained by the alophic plant showed a protective capacity against the UV radiation that was comparable to that observed with the use of a synthetic antioxidant compared to cells that were not pre-treated and then exposed to UV radiation. So this confirmed the potential use of these extracts in various fields such as nutraceutical, cosmeceutical, and pharmaceutical. Regarding the extracts obtained from microalgae, the evaluation of their bioactivity was preliminarily carried out in the laboratory where I conducted my PhD in an experiment where Sparosaurata fibroblasts were pre-treated with two synthetic antioxidants, beta-carotene and gallic acid, and then exposed to a mixture of emerging contaminants, BDE47, cadmium chloride, and carbamazepine, to assess their protective capacity against oxidative stress. On the basis of these preliminary data, in the course of my PhD studies, I evaluated the in vitro bioactivity of the extracts that were obtained from Nanochloropsis, Isochrysis galbana, Phaeodactylum tricornutum maintained both under standard and under nitrogen deprivation condition. Then C-brim fibroblast cell line were pre-treated with those extracts and then induced to oxidative stress with a selected chemical inducer with known pro-oxidant power, that is the BDE47. The pretreatment of the cells with the natural extracts showed the same protective capacity as the synthetic antioxidants against the chemical inducer BDE47. Furthermore, as it is known that microalgal extracts can have bactericidal activity, the bactericidal activity of the extracts obtained was tested, and in particular, Phaeodactylum tricornutum showed the highest bactericidal activity at the lowest concentration of the extracts against two bacteria that are Vibrio arvei and Vibrio anguillarum, that are two fish pathogens that are relevant in the aquaculture sector. Therefore, the use of microalgal extracts could find application not only in the nutraceutical, cosmeceutical, and pharmaceutical field, but also as a replacement for antibiotics. So in conclusion, we can say that this PhD project has highlighted that integrated multi-trophic aquaculture can guarantee high levels of productivity in the aquaculture sector, and that the co-cultivation of extractive species can ensure nutrient recycling and biomass production. This biomass is rich of bioactive compounds that have antioxidant and antimicrobial properties that can find many applications in different fields such as nutraceutical, cosmeceutical, and pharmaceutical industries. Thank you.
Fantastic. Thank you very much, Eleonora. That was a very good presentation, very clear, and very, very interesting results. So you've done a lot of work during your PhD. You should be very proud of yourself. And I would like to myself and Concetta, we can share this session by asking questions. But before we do so, I'm going to read quickly Eleonora's short biography, and then that'll give you the time to, if you want to type a question, you can type it on the comments box, or you can just raise your hand and we can take questions in an order. So I'm going to start now. So Eleonora, have you defended? Are you a doctor or?
Yes, I am.
Dr. Curcuraci then is a marine biologist, has a PhD on Earth and Marine Sciences at the University of Palermo. Currently, she is a grant holder within the Marine Hazard Project, funded by the Italian Ministry of Research, and a scientific supervisor for the research group of the Marine Biology Institute of Trapani within the Italy-Tunisia project on marine biotechnology, ARI Biotech. Her research mainly focuses on marine organisms, cultivation, and their cultivation for use in integrated multi-trophic aquaculture systems, as we have just heard. And I think someone else wants to get in the meantime. For helping biomass production, value-added natural compounds production, and inorganic and organic nutrients removal methods like bioremediation. Use of model marine organisms for bioactive molecule productions in response to environmental stressors is another topic that she's working on, as well as evaluation of effects of contaminants on marine model organisms by ecotoxicological, biochemical, and molecular analysis. Currently, Eleonora is a member of the board of our board in the European Society of Marine Biotechnology as a student representative and responsible for social media of the European Society, and she is a member of the Italian Association for the Study of and Application of Microalgae, which is called AISAM, and a member of the Italian Society of Experimental Biology, SIBS. I hope I pronounced everything correct, Eleonora and Concetta. And with this, I hope I gave you some time to think of your questions, write any comments, and yeah. So
as part of the board and as responsible of the social media divulgation, I would like to invite all the participants to follow us on LinkedIn, on Instagram, and all the social media, and also our website. Since
yes,
we do many activities, I can confirm that we do a lot of things, and they can have a lot of information from our Instagram page or from LinkedIn. So they are invited to follow us if they want to join the society.
Correct. And also we will be uploading this presentation later today. I'm gonna include the questions, if we have any, into the recording, and then I'll stop the recording. So Concetta, would you like to say a word or two?
Yes, thank you very much. Thank you, Maria, for organizing with us this webinar, and thanks, of course, to Eleonora for talk. And I would like to thank also the participants attending the webinar. Among these, we have colleagues, young researcher, or participant project, and some of our students that I want to thank. But first of all, I want to suggest them to have a look to the profile of our society, as the experience of Eleonora. It could be a suggestion for a young researcher, for student, master student, PhD student that want to enter in an international community of the marine biotechnologies. As each year we have the possibility to host students for her thesis, for project, for training project in the laboratory that belong to our network. So this could be a very beautiful experience for our students that want to enter in the international environment. And Eleonora is an expression of that success, as she grew in our lab. She did her PhD with us, continuing to join the society. So I hope that her experience could serve as a suggestion for other young researchers that want to enter in our community. I don't see question, but I hope that someone can be can have some curiosity or something to ask, we are available for you. And in any case, there are yes.
I have a question, a couple of questions. It's very interesting work, Eleonora. I'll open the questions. Please, guys, feel free to ask anything you found interesting or even to comment on something. Just for me, I'm very always interested in microalgae, also macroalgae, but so very interesting the fact that you found the biocidal impact, bactericidal impact from Phaeodactylium tricornutum. And by the way, huge kudos for pronouncing all the species very correctly and very understandably. This is not always easy when you present. So is it, how did you start thinking about using the extract as a bactericidal? Is it other studies that suggest that? And are you thinking of testing other species for this?
Yes, always everything starts from the literature. So there are some other examples on extracts of microalgae that can be used in this way. I have to say that all the extracts that we tested had bactericidal activity. I only showed the ones that we had from Phaeodactylium tricornutum that was maintained under standard condition, just because was the highest bactericidal activity with the lowest dose of the extracts. But all of them had the same capacity. Yes, of course, we should implement the species that we should analyze in order to find out if some other strains have the same capability, maybe using a lower dose than the one that we already tested. Thank you for your question.
Very interesting, and also so much more sustainable than biocides, which is a huge upcoming issue. Well, it's existing issue in aquaculture. And maybe I was thinking maybe a mixture of species of extracts. Could that be something, or has it been looked at?
We never tried, but it's a good suggestion. So maybe we can try, and I will let you know if we will have good results or even not.
Yeah, but I'm sure it will be, if it works for one, and then maybe a combination would be interesting. Before I move on with my second question, does anyone else has something to add or ask? Okay, I'll take the floor again. So Eleonora, you work then, as I can see, with the industry quite a lot.
Yeah.
Because it's an interesting aspect, working academia with industry for these important questions. My question is very simple. How did you find the experience?
I think it was a great experience also because you have the chance to see things from another perspective. We only have the perspective from the scientific side, but sometimes we miss the economic side. So we need to find like a balance in the middle. So it's not only what we think it's the best, because maybe it's impossible for an industry to achieve that goal from an economic point of view. So it is important to understand which are the needs of the company, first of all, and then science can be a support for its implementation. Thank you.
Great answer, Eleonora. That's what I thought also, that whenever working with the industry, it's interesting to see the implementation and actually asking the right questions for their needs anyway. Any other questions?
May I do just not a question, but asking please to Eleonora to, along with the explanation of the benefits collaboration with enterprise, the example related to the study of the internal metabolisms of the macroalgae set up in the laboratory. Can you please explain the benefit at lab scale, which is the benefit to study at lab scale the metabolic response of microalgae in order to set up the best culture condition, because this was an aspect, a new aspect that Eleonora studied during her laboratory. So she studied both the productive phase, but also the metabolism by gene expression. So if you could explain this as a perspective of study for new students.
Yes, it is very difficult to understand the biochemical aspects of the microalgae. And this aspect gave us the chance to understand which is the gene expression of specific genes that are correlated to oxidative stress. So this can give us the chance to better understand the quality of the culture that we have in the laboratory scale, and then to transfer this information to the industry in order to implement the quality of the culture that we are working on and that we are culturing, of course.
Thank you very much. And of course, before to translate the culture to the industry, we needed to set up the best condition in the laboratory. So part of the work is also this, is a very nice perspective of scientific exploration, especially for the research.
Absolutely, I totally agree. And I have one more question, Eleonora, on top of what you just mentioned with Concetta. So if you say you discovered with certain genes that you can understand the quality of your cultures, then if it's set up not in a lab, but in a more in an industrial setup environment, would you still go out and provide them, of course, the best conditions that gave you the best results, and that's your input into this? Would you still monitor now and then the quality? That's that with the, you know, by targeting the certain genes, would that help the industry to, for example, at certain intervals, to see if their product is as good as expected?
Yes, it can, and I think it gives us an information before. I mean, if you analyze this at the gene expression, so you will have the possibility to modulate the culture in order to implement the quality of the culture that we want to obtain, and you can remodulate the cultivation conditions in order to obtain what you want to obtain. So it's a preliminary step. It's
I understand. Perfect. So I personally don't have any questions. Well, I have more, but you know that I can ask you, Eleonora, at another point. And obviously, everybody, maybe Eleonora would like to share her email, or
yes,
you can contact us. Yeah, you can write your email.
I can write it here.
At this point, I'm stopping the recording. So it's not a huge file, so I'm stopping it.